So our speaker today is Dr. Logan Spector. Logan is a professor of pediatrics, and he is the head of our division of epidemiology and clinical research. Dr. Spector is a graduate of the College of William and Mary, and then went on to complete his PhD in epidemiology at Emory University in Atlanta. Logan joined the University of Minnesota as a postdoc in 2002, joined the faculty in 2003, and worked his way up through the ranks and was promoted to professor in 2015, at which time he also took on the leadership of this division. Logan has multiple accolades. He serves on multiple NIH study sections. He serves on study sections for cancer epidemiology, essentially around the world. He also holds the Suzanne Holmes Hodder Chair in Cancer Research here at the University of Minnesota. He's an outstanding mentor, leader, as evidenced by the success of his mentees and the longstanding training grant that Logan holds in cancer epidemiology. So with that, I'd like to thank Logan, and we're very much interested in hearing what you have to say this morning.
Thank you, Joe. So my title today is Can We Prevent Childhood Leukemia? And fair warning, I have a long history of giving talks grandiose titles that I can't quite deliver on, but I hope you'll stay and listen about the state of the science answering the question. And I will say that, you know, as an epidemiologist, I've been satisfied for the last 20 years with description of childhood cancer and especially leukemia risk. But lately I've been posing this question to myself and wanting to retire having answered it satisfactorily. So full disclosure, I have nothing to disclose. And learning objectives today are one, to understand current ability to predict risk of childhood leukemia at birth, and two, to recount modifiable pre and postnatal factors for childhood leukemia. Joe already mentioned that I head the Division of Epidemiology and Clinical Research. We are larger than cancer, so you may know doctors Erin Kelly and Justin Ryder, who study childhood obesity, and Sarah Cusick, who studies nutrition and cognitive outcomes. But I'd just like to highlight my faculty that concentrate on pediatric cancer. Aside from myself, there's doctors Jen Pointer, Erin Marcotte, and Lindsay Williams, and I'll be highlighting some of their work during the talk as well. And our division is mission-based, the mission being understanding the causes of childhood cancer in order to predict who'll get it, to enable early detection, and someday to enable prevention. And because we're mission-based, we take all the possible options for studying etiology. So this includes surveillance data at the national and international level, record linkage studies, which we've done in Minnesota nationally and through pooling efforts internationally, conducting case control studies through the Children's Oncology Group, and then more recently, the Childhood Cancer and Leukemia International Consortium, which I head and which I'll describe later. And then more and more genomics. So that includes genome-wide association studies, whole exome sequencing, whole genome sequencing, and I'm leaving out a lot of other omics that we're employing. The last mode of analysis is becoming so important that I've formed the Childhood Cancer Genomics Group, which consists of me and our cancer faculty, but also drawing in collaborators from around the university, including from epidemiology, lab medicine, biostatistics, and then a shout out to my colleagues in P.T. Monk, Bo Weber, and Brandon Moriarty, who help us with post-GWAS or post-sequencing functional validation. And we also have two fantastic bioinformatics and genomic analysts, Doctors Lauren Mills and Andy Radusky, that help us keep analyses moving. So obviously, the major factor driving most of what's possible in pediatric cancer is its extreme rarity. So in the pediatric age range of 0 to 20, about 1% of the total cancer burden is diagnosed. And within childhood cancer, of course, there's significant heterogeneity in the types of cancer that are diagnosed, including by year of age, attained age. But you can see in blue that leukemias are the largest share early in life and then decline later. Making this challenge of understanding etiology even greater, there's very significant cytogenetic or diversity in somatic genetics here in B-ALL and here in AML as well. And because we now have evidence for what we call etiologic heterogeneity, meaning that the causes of leukemia can vary by specific subtype, this means that our work is far from done in determining the causes. I should add that etiologic heterogeneity doesn't mean that causes are completely separate, but that the degree of association may differ, and in some cases, risk factors may be unique to particular subtypes. In our division, we've slowly been attacking the causes behind long-known demographic risk factors for ALL. So here we looked at the sex ratio, male to female, in ALL and AML, among other cancers, by single year of age, reasoning that lumping together 5-year age groups in pediatric cancer is obscures signals that we might find, knowing that children can change in the space of a year very easily. And so we do see that the sex ratio changes quite a bit. In fact, it gets noticeably lower right at the prepubertal dip in growth. We also are trying to find out, is sex just a proxy for other exposures or other variables, or is it in and of itself molecularly related to risk of cancer? And what we found is that no matter what we control for, so for instance, boys tend to have a higher birth weight than girls, no matter what we control for, the estimates are about the same, suggesting that sex is actually biologically, molecularly somehow driving differences in cancer risk. There are also persistent racial ethnic differences in the rate of especially ALL. AML is much less stark, but noticeably in the US, I guess I don't have a legend here, but the blue is white children. The dashed line, gray line is Asian Pacific Islander, and the purple line is African-American children. And they have Latino children on a different graph, but they have about 25% higher risk than white kids. And this pattern is also replicated internationally. So for these reasons and others we don't have time to get into, we think that the difference in rates, because they are consistent between originating countries and diaspora populations, are probably genetic in origin. And these are being examined in multiple places, including Minnesota. All right, so the demographic risk factors are fairly clear. We also have good evidence that pediatric cancer starts in utero. And it comes in two flavors. First, we have circumstantial evidence, including the young age at onset. So the peak age for BALL is from 2 to 5. And if you think there's an induction period, then that starts to place you easily into gestation. In the solid tumors and maybe liquid tumors as well, histologic resemblance to embryonal or fetal cells. And then we have some proof of principle in solid tumors. There are case reports of prenatal diagnosis by ultrasound. And then we have the fascinating story about leukemia in twins and backtracking. And this is all enabled because leukemia, of course, is a circulating tumor, which also I think is what makes the possibilities for prevention much more feasible. So Professor Sir Mel Greaves pioneered this field of study. He noted that he collected over a period of years identical twins with leukemia, and noted that depending on the age they were diagnosed, there could be either exquisitely short time between twins being diagnosed or fantastically long. So in one case, one twin was diagnosed at 5 and the other at 14, whereas among infants, the time between was in a matter of weeks or months. And the hypothesis behind this is that we know that monozygotic twins, 60% of them share placental vasculature, so that blood can travel from one twin to the other. And here you have it depicted in an old painting of the Dutch painting called The Swaddled Children, depicting twin-twin transfusion syndrome. And sure enough, when looking at it molecularly, Professor Sir Mel Greaves found that the twins tended to have not just the same type of leukemia, but the same translocation with idiosyncratic breakpoint, thus demonstrating that in all likelihood, the leukemia started in one twin and then spread to the other during gestation. And so that was maybe highly suggestive circumstantial evidence. The nail in the coffin was finding patient-specific translocations in birth samples, meaning dried blood spots or cord blood. So here's an early example of that. So we call that backtracking. And in a recent review paper from April, we recount this and suggest that this area of inquiry be revived and considered for application to understand the natural history prospectively of leukemia, and someday, not to alarm the newborn screeners, but to start to think about, can we screen for leukemia risk? So I will add that most of the work has been in ALL for fairly obvious reasons. It's far more common than AML, but we do have a smaller but suggestive set of evidence that AML also starts in utero. So in this review, we looked at the particular translocations or aneuploidies that have been backtracked in pediatric leukemia. And you can see that the percent of birth samples that are positive for patient-specific mutations varies, but has a pretty good pattern where it's about 20 to 25% are detectable if they have ETV6 RUNX1, formerly known as TEL-AML1, and more so in kids with high hyperdiploidy, and with a fair amount of consistency in estimates across the large range of countries where this has been attempted. More provocatively, people have for a long time tried to see whether leukemia typical translocations, or in some case aneuploidies, can be found in birth samples from healthy or unselected newborns. And mainly people have looked at ETV6 RUNX1 because it's the most common translocation in ALL and the most well characterized. And you can see that estimates range from 0% up to 5%. But I want to note that the technology has changed quite a bit over time. So most of it was RT-PCR, which is potentially prone to contamination. More recently, a German group has come up with a sequencing-based technology called GIPFEL, which presumably is more reliable and less prone to contamination. Nevertheless, GIPFEL is a pretty clunky assay, which could never be deployed on a mass scale. For one thing, it requires a very large input that you'll only get from cord blood rather than dried blood spots. And I think that dried blood spots already being collected on nearly every child are the preferable medium to attempt screening. And so colleagues, Dr. Aaron Marcotte and Heather Nelson have sent up the BAT signal. And my only contribution to this work, by the way, was the acronym. So I'm really touting them. So I did come up with breakpoint agnostic translocation screening, or BATS. And the point here is to take a punch from a Guthrie card, a newborn blood spot, and apply ddPCR, or droplet digital PCR, which is a method for detecting very low copy number segments of DNA. And I will let them explain in another venue how it is they came up with primers that allow you to capture most ETV6 RUNX1 translocations across the range of breakpoints. But this has been a fairly successful pilot, which is poised for publication and expansion. So I'll just show you very quickly. So in cell spiking experiments, that is diluting ETV6 RUNX1 positive leukemia in increasingly large pools of normal cells, they achieved a very good correlation between the input and the ddPCR detection. And in blood spots that we had from ALL patients, unfortunately without cytogenetic information, they did find 45% were positive for ETV6 RUNX1, whereas among unselected newborns, they found 15% were positive, which is 3 times higher than the highest estimate, but still not unexpected to find ETV6 RUNX1 in some share of newborns. Now I'll talk a little bit about my new project with a grant that literally started this past Sunday. We're calling Record. And the critical questions we'll be answering in this one are, what subtypes and in turn, what proportion of childhood leukemias develop in utero? So I previously showed you a list of the subtypes that have been backtracked before, but that leaves a very long tail of translocations or aneuploidies or other somatic features that have not been backtracked. Secondly, what is the specific cell of origin in which pre-leukemic clones arise? This has been, people have attempted to answer this for many years, from phenotypic comparison of leukemia cells to normal cells and mouse models, et cetera. But we plan to look for specific cell types with cell sorting and looking for the population with the greatest density of pre-leukemic clones. And then lastly, are some newborns more prone to developing leukemia-initiating lesions than others? In other words, do we see more pre-leukemia in kids that have the risk factors that epidemiologists have defined over the past 30, 40 years? And this is all enabled by the Children's Oncology Group Project Every Child, of which I am a committee member. my contribution there was to get a upfront questionnaire of newly registered and diagnosed children. And I snuck on a question about whether cord blood was banked at birth. We were pleased to see that several hundred children with leukemia have already reported having banked cord blood. And you might ask, what if they need it clinically? And for the reasons that I've mentioned before, namely, we think a large proportion of them have circulating pre-leukemic clones. I think these cords are taken out of circulation for clinical use for fear of reimplanting a leukemia. And so we also, word of this study has gotten out to some patients, and they're actually asking us to donate their child's cord blood because it relieves them of the maintenance storage fees. So for this grant, we will focus on non-aneuploidies, mainly because developing primers for translocations or other subchromosomal events is a lot more straightforward than developing an assay for finding low frequency circulating aneuploid cells. But that is 3/4 of ALL and 2/3 of AML. And this is a complicated diagram of the study, but basically we hope to end up with about 50 AML cords and 200 ALL cords, and we'll focus on roughly 180 non-aneuploid children, and we will do whole genome sequencing and RNA-seq on the leukemia cells, and from that develop patient-specific primers, which we will screen in cell-sorted cord blood. For those that are positive, we will select a select few for single cell analysis and what we're calling the deep dive, and we'll be using something called Target-seq, which was developed by our collaborators at Oxford. And Target-seq is simultaneous DNA and RNA sequencing at the single cell level. And so what we'll gather from this is multitudinous. But for the first time, we'll have identified evidence of the cell of origin in an in vivo example. Secondly, we will know what the difference is in transcriptome between partially mutated and fully mutated and leukemic cell. And there's the opportunity then to study the biology of pre-leukemia, which we hope would identify some vulnerability to chemoprevention. So I was chatting with Joe before you all were let into the room. He said, well, what are you going to do if you can predict who gets leukemia? Give them a dose of prednisone. And I said, well, that's not entirely out of the question. Maybe we could get away with a much lower dose. And once we know what the vulnerabilities of pre-leukemia are, I'd say that, then we are off to the races. So that's it for where we are with screening for signs of leukemia. But we also have additional demographic, genetic, or environmental risk factors. And this really is what I've spent the last 20 years of my career defining. So, is it environmental exposure? Is it DNA? What's the role of just sheer chance? We'll start with DNA. So sequencing has revealed what we sort of already knew from familial patterns in pediatric leukemia, that only a small proportion of leukemias have a pathogenic or likely pathogenic mutation in the classic cancer susceptibility gene. And this is not to say that there aren't more out there. Or some that are associated with specific subtypes. So in, almost 8 years ago, with colleagues at WashU, we published on an exome sequencing study in MLL negative infant leukemia, which is phenotypically distinct from leukemia at older ages. And we looked at, classic and canonical and some non-canonical cancer susceptibility genes. And this diagram is maybe a little bit obscure without explaining it, but basically it's a statistical test for the red dot is the distribution of pathogenic or likely pathogenic mutations in MLL family genes. In other words, in germline, not in the somatic tissue, compared to the expected distribution. And you can see for AML, it's way out of whack. What's interesting is that it's not a single gene in the MLL family that's mutated. It is one of several, and for many of these kids, it was an example of digenic inheritance, meaning one version of MLL has a heterozygous pathogenic mutation, and another version of MLL has one. And this gives us some inkling, and let me add that there's similar evidence as yet unpublished in MLL positive infant leukemia. This gives us some inkling that we may have a much stronger genetic predictor of risk for infant leukemia specifically than we hope to achieve in leukemia at older ages. I should add that there's been a small cottage industry in the last few years of finding new moderate penetrance familial predispositions to leukemia, many of which involve germline mutations in genes that are commonly somatically mutated in pediatric leukemia. So here's Icaros or IKZF1 germline mutations predisposing, but there's also been syndromes identified with PAX5 and ETV6. And I fully expect that if we surveyed all the genes that are involved somatically in pediatric leukemia, that we would probably find more syndromes like this. But surprisingly, common variation, otherwise known as SNPs, meaning variants that are present at greater than 1% minor allele frequency in a population, are strongly associated with risk of ALL. And so this was should have been the lead diagram in this paper, but it was buried in the supplement. It's simply showing the distribution of risk according to polygenic risk score for roughly 18 SNPs that are associated with BALL. And it's a little bit deceiving because the referent here is the median of the polygenic risk distribution. And the people at the highest risk have roughly 5-fold. But if you compare to the bottom of the distribution, it's actually describing something like a 10-fold difference in risk. And from an earlier iteration of this graph, I once calculated that 90% of leukemia comes from the upper half of this distribution. And so another indication that with genetics, we may be able to push risk prediction in a productive direction. I'll add more recently, this is in preprint, but this was a analysis looking at in UK Biobank, what are the SNPs associated with hematopoietic traits, such as the basophil and lymphocyte, neutrophil level. And then here we have lymphocyte to myeloid cell ratio. And these graphs are different modeling techniques. But what you see is that there is an increased risk by about 23% for a 1 unit change in the lymphocyte to myeloid cell ratio, suggesting that genetic propensity to higher lymphocyte count, especially in relation to myeloid cells, it increases the risk of ALL. And it hasn't been applied yet to AML, but you can imagine that the inverse would be true. And I also mentioned this, although it's not a large effect, that we can plumb the genetic data for more associations with traits that we think are related to leukemia and help push the genetic prediction even further. As far as non-genetic risk factors for leukemia that are accessible at birth and which we might put into a risk prediction algorithm is birth weight, which here we looked at not just according to immunophenotype, but by cytogenetic feature, and showed that the association varies. I should add that these relative risks look pretty small, but these are per 500-gram increase in birth weight. So across the normal range of birth weight, we're actually looking at more like a 50% increased risk. There's also been a fair amount of studies looking at parental age. And so this panel is maternal age, but there's a similar association with paternal age. And since those tend to be highly correlated, we can't easily tell whether it's mother's or father's age or really both. But what we see is about a 5% increased risk per 5-year age difference. So if you think of the usual range of reproduction is about, in mothers at least, about 20 or 25 years, then we're similarly looking at roughly a 50% increased risk. I'll add that Dr. Marcotte in my division pulled off a very interesting analysis looking at C-section, which originally we thought would be itself the potential risk factor because we know microbiome can differ depending on route of delivery. But interestingly, there was no association with C-section alone. But when we looked at pre-labor C-section, there was a slight, almost 25% increased risk of ALL, but not AML. And that got us thinking the microbiome is no different between a pre-labor and a C-section with labor. But the major difference is exposure to labor itself, which I've been told is very stressful and witnessed. But the fact is that children who have not gone through labor have been demonstrated to have lower levels of cortisol. And so we hypothesized, let me back up, glucocorticoids are a backbone of ALL therapy and are essentially synthetic cortisol. And so we hypothesized that exposure to labor has a culling effect on pre-leukemic clones. And backing up a little bit to mention bats, we can actually now test this with the right data set. Also suggests that it may be possible to modify the population of pre-leukemic cells. And then more tenuously, but interestingly, there's a suggestion that children at birth, this is in blood spots, have differing levels of cytokines than children who don't go on to develop leukemia. And then I'll add there's whole classes of technologies that we haven't deployed widely to try and predict who might get leukemia. So for instance, DNA methylation is very feasible to use in dried blood spots and could be deployed to come up with a risk prediction classifier. So to summarize, we have common alleles. I'm counting both those found in GWAS for ALL and the trait-associated alleles, they describe something like a 10 to 15-fold increase in risk. Moderately rare medium penetrance alleles describe a 2 to 3-fold increase in risk, each one by themselves. Rare alleles, describe a 20-fold increase in risk or greater. We do have protocols for screening children with high penetrance cancer mutations, syndromes, and I would maybe take them out of the mix here. Birth weight and parental age both increase slightly across the normal range. Pre-labor C-section, also obviously obvious at birth, could be added to the mix. And then as yet untested, if we combine the genetic predictors, the demographic, sorry, I should have added sex, and until we've isolated the potentially causative genes, race, ethnicity into this algorithm, we'd like to test those genetic, demographic, and perinatal risk factors in conjunction with pre-leukemia detection to see just how far can we push risk prediction. And I don't have an answer for you yet, but it's clear that we have all the ingredients to bake this cake. And now we need to pull the data together. So what could we modify postnatally to prevent childhood leukemia? So again, Professor Sir Mel Greaves, who started this ball rolling by proving that pediatric cancer can start in utero, thinks that we can potentially modify the infant microbiome. And his suggestion is that we come up with a probiotic cocktail. The best I could come up with is a yogurt lassi, but that we would give population-wide to put these kids on the right track in terms of microbiome. I'm personally skeptical that that we could pull off a population-wide microbiome manipulation, and think that we're better off finding the kids at the highest risk, or taking the highest portion of the risk distribution and attempting intervention with those groups. And so I'll just quickly recount what I think are the potentially modifiable postnatal risk factors. And so, we do have preconception and pregnancy risk factors, many of which run in exactly the direction that you'd expect. And we already, for instance, discourage smoking during pregnancy, encourage vitamins during pregnancy. I'm not sure what the guidance is on coffee, but I know that the guidance is no alcohol. And then, of course, we avoid giving diagnostic or therapeutic radiation during pregnancy, especially to the abdomen, for fear of causing leukemia. But we can't very well modify risk factors during pregnancy for a risk that is identified at birth. And so mainly we would be talking about postnatal modification of risk. And so among these, we have breastfeeding, allergies. There are some residential chemicals such as pesticides, which could be minimized. Some evidence for passive smoking, and then postnatal irradiation as well. And I just want to mention that much of what we know about what the risk factors are for pediatric cancer comes from the CLIC consortium, which I run, which has pooled studies containing more than 32,000 leukemia cases, allowing us to look at leukemia risk in very fine detail and very robustly and precisely. And one of our early observations was a fairly strong and time-dependent inverse association between breastfeeding and especially breastfeeding duration and lower risk of ALL. And of course, we promote breastfeeding as healthy as it is to all women, but it may be that for children who are at the highest level of risk for ALL, we could especially target pro-breastfeeding interventions. At the same study, we found a even stronger and inverse association between daycare attendance and especially the age at start of daycare attendance. As any parent and probably most pediatricians know, daycares are germ factories, and so it's not exposure to Eric Carle books that's doing this. We think that it is early exposure to germs that would train the immune system and suppress the risk of leukemia. We also took a look at farm residents and exposure to animals in early childhood. And on the left, we have exposure to livestock in the first year of life, which did have a suggestive inverse association. It differed based on the model if we used a random effect or a fixed effect. However, contact with pets, obviously most commonly dogs and cats, or in the first year of life, was associated with decreased risk in pediatric ALL. And here we have a potential intervention that many people could get behind. Puppies to prevent leukemia, I think would have high acceptance and would be potentially very feasible. Lastly, I don't mean to discount the possibility of remediating environment. As I mentioned, pesticides have a fairly small but robust association with ALL risk. And here we have a demonstration of association of air pollution, and in this particular case, estimated benzene exposure from air pollution on risk of ALL and AML. This also is an example of etiologic heterogeneity. So the risk in ALL is less and occurs more or less linearly with increasing dose, whereas AML has a higher degree of risk due to benzene exposure, and that risk sort of ramps up very quickly and then doesn't quite plateau, but grows more slowly. The point being that we could also mitigate the environment, or probably more likely move kids, suggest moving, that have the highest risk due to air pollution, or suggest that parents not use pesticides, etc. So I've come around to the posing the again, the question that I put in the title: can we prevent childhood leukemia? And I want to suggest that clearly the answer is not yet, but the end goal is coming into sight. So I think, first of all, that risk prediction at birth is rapidly advancing. And I myself am pursuing the studies to tie together the genetic risk prediction, the demographic, perinatal risk factors, and tie them into together with prenatal circulating pre-leukemic cell detection to see just how far we can push this risk prediction, and what share of the population would we need to target for preventive efforts to have the largest impact on the share of leukemia. Secondly, I think there's enough evidence, and I didn't talk about experimental models, but that immune modulation holds promise as an intervention. And whether that means giving puppies to kids or giving them a mango lassi remains to be seen. But either way, these are have potential evidence behind them. I have not mentioned, because there are no such studies yet, but they are enabled by the invention of bats. But we also could conceive of longitudinal monitoring of pre-leukemia and looking for signs of clonal expansion. And this is obviously enabled by the fact that it's a lot easier to get blood samples than solid tissue. And so without a doubt, this would first start in natural history studies, but we may learn something that allows us to make this feasible. And then lastly, chemo prevention is as yet undreamt of, but I mentioned the Recount study, which I hope will help identify vulnerabilities to pre-leukemia. And I mentioned the hints in the literature that some exposures may actually cull pre-leukemia, which we then could verify through detection or sort of analogous minimal residual pre-disease, if you will. So with that, I am happy to take questions, and thank you very much for your attention.
All right. Our first, I think you kind of alluded to this, but I'm sure people wouldn't mind hearing you talk about it a little bit more. So this comes from Sue Berry in our Genetics and Metabolism division. Dr. Berry says, very exciting work. What do we know about the pre-symptomatic phase of leukemia? Are there potentially babies who would test positive but not develop disease? How would one decide who and when to treat?
Yes, and Sue, I fully anticipated this question because from a newborn screening perspective, the ethos is that your test should be deterministic, not probabilistic, and you should have an intervention in place to prevent the poor outcome. As you know, that kind of firewall is breaking down in newborn screening. So we now screen for things where there's a deterministic test but no intervention. And I think the walls are cracking a bit because we now have maybe more probabilistic tests getting introduced that may or may not have an intervention. So I will say that for most forms of leukemia at older ages, we're not going to have anything close to a deterministic test. It will be probabilistic for the likely future. I think it's possible for MLL mutated leukemia that we will have something close to a deterministic test. This is because the association between pathogenic mutations in MLL family genes is very strong, and we think that the MLL mutation is itself causes leukemia without much need for cooperating mutations. Kind of a rare example of a single mutation that will cause cancer all by itself. And so, I've been pushing the risk prediction end, so I can't tell you what the intervention would be for infant leukemia, but the circumstances around infant leukemia are probably the most promising to move it towards newborn screening anytime soon. For other forms of leukemia, I absolutely agree. We're going to find many more parents, many more kids that have a degree of risk that aren't going to convert. And no, we don't know what the symptoms, if any, of pre-leukemia are. These cells are circulating at 1 per 10,000 lymphocytes, and so it's not likely to have any actual overt phenotypic effect.
I just want to clarify, because you're, I think, using terms in a way that the way the words mean, but I haven't heard people talk about it in that way before. So when you talk about a newborn screening test being deterministic, what you're talking about is the concept that when you do more newborn screen tests, yes, you might have a false positive, but once you've done the appropriate follow-up definitive testing, then you have the disease. It's determined versus
probabilistic. If
have it, is that the distinction?
If you're missing an enzyme, then you're going to have the phenotype if there's no intervention. In just think in the simple case of say Li-Fraumeni syndrome, even finding that mutation doesn't mean that you're going to get a particular form of cancer. There's still some other factors involved, and so this I perceive as the barrier to implementing newborn screening for childhood cancer risk.
One of the things I was curious about as you were talking about the things that seem to have fallen out as protective risk factors is just the influence of social determinants of health broadly, and things like zip code and household income and parental education attainment. What's known about those aspects as risk factors for leukemia?
Yeah, so we did a population-based study in Minnesota several years ago where we linked the cancer and birth registry. And our question was, how much of the SES gradient in risk is due to known factors? And surprisingly to many people, ALL is more, and in pediatric cancer in general, is more common the further you go up the SES gradient. But that gradient more or less disappeared once we controlled for birth weight, parental age, and race ethnicity. And so, Minnesota is not a large state, so it's possible that in a larger state we would, or larger sample, we would find a residual association of SES. But in Minnesota, it was more or less all explained by the social patterning of risk factors.
Thank you.
Well, if I've stunned the audience into silence, I'm happy to wait for more questions. But
yes, I think I always have lots of questions for people. I think that, just really was kind of thinking about this concept of, especially when there's been an older sibling or a family member who's experienced cancer, and that sort of balance of trying to give a family some idea of things they could do to promote the health and well-being of their child who's currently not affected. And, what could we say now? It sounds like we would say most of the things we would already say: eat a varied diet and try to breastfeed if you can. And so
I mean, the others have studied sibling recurrence, and unless there's a strong familial syndrome, there's not much recurrence risk, or nothing that an individual should be concerned with. So we can already give that reassurance to parents. Yeah.
Great.
And these are all things that we wish to promote, anyway.
Right.
Breastfeeding, not smoking, take your vitamins. So.
Yeah, but I think that's, we ask a lot of quote family history questions when we're taking in a new patient, but I think probably there are many of the things that we ask about that in fact happen spontaneously and are unlikely to be increased in a family member. So that's, at least for leukemia, it's reassuring to know that that doesn't seem to have increased risk for siblings.
Yep, extremely reassuring.
Great. Well, your group is doing incredible work on a number of fronts. So thanks for sharing your knowledge with us. And I think it, definitely just gives us food for thought about the kinds of things that we can be talking with patients and families about. What advice do you have for, general pediatricians on the front lines in terms of helping families to get involved with clinical research projects? I know that can be a challenge, sort of getting patients enrolled into studies that you may be doing. How can we drive people who are interested in contributing to finding good ways to contribute?
Well, I wish I had insight into that, Emily, because these days we're having to convince people to take vaccines. So being in clinical studies is, often gets called a guinea pig. I'm not sure if we can't get them to take vaccines that we can convince them to take part in studies. A lot of our work is now using biobanks and population data that's available to researchers, and so convincing people to participate isn't so much an issue for us epidemiologists. I think that we have a political science problem, which is why do some people not trust the medical establishment or science in general, and how can we bring them back to have that trust? That's somebody else's job to figure out.
Yes, that would take us well past our 8:30 time slot, but yeah, I think the big data and all the things that are available to us to help draw conclusions at a population level is pretty fascinating these days.
It is.
Well, thank you so much for sharing your knowledge and expertise with us this morning. Everyone have a great Wednesday morning. Enjoy August, which is here to shine some sun on us. You can see coming in through my window. Have a great day, everyone.
Thank you.